3' untranslated region. And more, it was characterized that it could regulate mRNAs involved in inflammation like TNF-alpha. And so this RNA binding protein is indicated here. It's a member of a small family of these zinc-finger proteins. It acts as a post-transcriptional regulator. Very interestingly, if you take a mouse and delete this TTP protein from its genome, it develops severe inflammatory diseases such as cachexia, dermatitis, and arthritis. And it's presumed that this is due to, or it's been shown that this is due to overexpression of COX-2 and TNF-alpha and other inflammatory mediators. So basically, this protein will bind to RNAs like COX-2 on this signature sequence and promote their decay, thus limiting their expression. And here's just an experiment illustrating how this TTP protein can regulate COX-2 expression. We engineered some cell lines here where we could induce TTP expression in those cell lines using a tetracycline-inducible system. And in this particular cell line here, you can see where COX-2 is overexpressed and TTP is present. It robustly downregulates it. So how does TTP make this happen? Well, in other data, the mechanism of TTP's ability to regulate ARE-containing gene expression has been shown in that TTP will bind to these mRNAs and bring these over to little cytoplasmic bodies in the cell that contain RNA degradation machinery. And these bodies are called P-bodies. And there's a number of different components involved with degrading mRNAs that are present in P-bodies. And you can see these P-bodies doing some either transfections of GFP-tagged components or by doing some immunocytochemistry. And you can see these little dots in the cell. That's where RNA degradation is occurring. And TTP brings mRNAs like COX-2 to these dots so that their expression can be controlled. And this is just illustrating this. I don't know how easy this is to see, but if you stain cells for a P-body marker or a P-body component, one this decapping protein called DCP1A, you can see a lot of dots. You stain the same cell with TTP protein, you see similar numbers of dots, and when you merge these two images together, where you can see yellow on here, you see the co-localization indicating that TTP protein co-localizes with mRNAs on these P-bodies. Okay, now I'd like to spend a little bit of time talking about the opposite RNA binding protein called the HUR protein. Now, this protein is a member of the Drosophila elab family of genes. 4 members in this. 3 of them are primarily expressed in neuronal tissue, whereas the HUR protein is primarily expressed in the nucleus of cells. Now, this is an important component because when HUR becomes overexpressed in a tumor cell, as I'll show you, it gets trafficked out into the cytoplasm. And the cytoplasm is a place where RNA stabilization occurs. And so the cell appears, as I'll show, appears to have a problem with keeping the tumor cell appears to have a problem of keeping HUR in the nucleus. And normally, HUR serves a nucleocytoplasmic shuttling function where it will take mRNAs like COX-2 or TNF-alpha when they're expressed and aid in their transport from the nucleus to the cytoplasm. And then it shuttles back into the nucleus. It can bind these A-rich mRNA elements. And when HUR is overexpressed in cells, it acts to stabilize these mRNAs by binding to this signature sequence. Now, the exact mechanism how that occurs is not entirely figured out yet. may have a microRNA involvement component to it, maybe just a protection from this mRNA from endo and exonucleases, and then it also may be by keeping this particular mRNA out of P bodies where the degradation can occur. And here's another example using a similar cell line where we could induce the overexpression of this HUR protein, and that's shown here using a tetracycline inducible system. And in these cells where you induce HUR overexpression, you also promote COX-2 expression. And so again, this is acting the opposite as that TTP protein. And I've been primarily showing you just a target gene, COX-2, but remember, these AU-rich sequences, which these proteins bind to, they exist in 5 to 8% of all of our genes. And so you would expect that, well, if COX-2 is overexpressed, potentially another ARE-containing gene may be overexpressed. And working with collaborators at Vanderbilt Medical Center, we looked at expression of ARE-containing genes globally in normal colonic epithelium compared to late-stage colorectal cancers. And in a nutshell, we found that these ARE-containing genes were now upregulated to a point of over 20% of the transcriptome, indicating that these particular mRNAs are stabilized. And so then that begs the question, these factors which control the expression of all of these genes, what happens to them during tumorigenesis? And so we went ahead and paneled, at least shown in here by RNA analysis, we paneled normal versus stage colonic tumors, and this is just indicating graphically what the tumor staging might indicate. And shown over here is TTP gene expression. We can see very robustly here that TTP mRNA expression in the colorectal tumors is lost. And I'll show you some histology validating this. Just the opposite, HUR expression in normal tissue is moderately low, whereas it becomes upregulated from 2 to 5-fold increase in the HUR mRNA in the colorectal tumors. So what we have here is a change. In the normal tissue, the RNA degrading protein, TTP, is elevated, and the HUR protein is downregulated, or is at a low level. Whereas in the tumor tissue, you see the opposite effect occurring. And when we go ahead and stain normal, adenoma, or polyps, and adenocarcinomas from colon cancer, again, focus on the brown staining here, TTP expression is going away. And especially in the adenocarcinoma, it's very difficult to see any TTP expression. Just the opposite with HUR. In the colonic epithelium here, it's present only in the nucleus at modest levels. In adenomas, it becomes overexpressed, and you can start seeing here it's leaking out into the cytoplasm of those particular epithelial cells. And in adenocarcinomas, HUR is abundantly overexpressed. So again, we have this switch in overexpression of the RNA stabilization protein and loss of expression of the RNA decay protein. So now I want to switch things just a little bit here and talk about some more recent work we've done in the lab, asking a question. Well, we know in tumors this RNA stability factor, HUR, becomes overexpressed. Now, what purpose does that serve during tumorigenesis? And could potentially this RNA binding protein be an oncogene when it's overexpressed? And so what we created was a transgenic mouse here that would overexpress this HUR protein only in the gastrointestinal epithelium. And this is just an illustration here of the transgene construct. It's driven by an intestinal-specific promoter that will only express in the colon and the small intestine, and it's called the A3 promoter. It's driving the expression of a bicistronic construct that will make HUR flag and also GFP expression. Over here is just an RNA analysis showing that this particular RNA generated from this construct is only made in the colon and in the small intestine. Other tissues such as heart, kidney, spleen, liver, or stomach do not express this, as expected. Shown over here is looking at the transgenic mouse and the wild-type mouse littermate. And here is looking for GFP fluorescence in the small intestinal tissue, shown over here in these epithelial cells that we can see GFP being expressed. In that normal tissue, or from a wild-type mouse, we don't. Looking in the colon here and staining for HUR levels, we can see now here higher levels of the HUR protein as compared to the wild-type mouse. And that's due to overexpression of that particular transgene. Now, the one thing that was a little bit disappointing in making this mouse is that it didn't appear to have any abnormalities in the GI tract. These mice bred fine, they ate fine, they spent up my grant dollars quite nicely without any tumorigenesis appearing to occur. And so then we asked a simple question whether or not, well, these things act on RNAs, and these RNAs potentially aren't there at an abundant level where they have something to facilitate or something to stabilize. So we went ahead and we took these mice and put them into two tumor-initiating backgrounds. One, we modulated their genetic background by crossing it with an APC-Min mouse, and I'll explain a little bit more about what that mouse is. And then we gave this mouse inflammatory bowel disease, or ulcerative colitis. And we asked whether or not tumorigenesis would be potentiated in these particular backgrounds. And so the APC-Min mouse, it's a model of gastrointestinal tumorigenesis. These particular mice will get a high number of adenomas in their GI tract, primarily in their small intestine. You can quantitate the numbers and the sizes of these tumors. And this is just taking the small intestine here, staining it with methylene blue, and you can see the tumors coming up here. And these mice are a commonly used model, or in vivo model. They are mice that have a dominant heterozygous mutation in the APC tumor suppressor gene. The other allele is wild type, but in these tumors here, due to loss of heterozygosity, the wild type allele has been lost. And this is a model of familial adenomatous polyposis in humans. And so, as I mentioned, normal mice at 15 weeks of age, or these HUR transgenic mice, don't display any enhanced tumorigenesis. And here is just what I showed you earlier, a section of what an APC-Min mouse area of their small intestine would look like with these tumors. Now, when we take this mouse here and put that into an APC-Min mouse genetic background and look at tumor burden, you can see much more numbers of tumors being shown. And this is just two independent transgenic founder lines. And also, these tumors appear much larger. So this is indicating that HUR, when being overexpressed in this genetic background, can potentiate tumorigenesis. And we can quantitate this and show that you see about a 2 to 4-fold increase in the numbers of tumors that are present in the small intestine and in the colon, resulting from HUR being overexpressed. when we cut out those tumors and stain them for this HUR protein. In an ABC-min tumor, you can see some staining. In a transgenic tumor, overexpression of that HUR protein is everywhere in that tumor in the epithelium. A little bit on this ulcerative colitis model. So what this model basically is a mouse where you give them a procarcinogen called oxoxymethane here, or AOM, and you inject that into the peritoneal cavity of the mouse, wait a week, and then you give the mouse an agent in their drinking water called dextran sodium sulfate. It's an inflammatory agent which will give them ulcerative colitis. And then you wait. And in C57 black 6 mice, shown over here, these mice get about 5 or 6 colonic tumors in their colons here. And whereas if we do that same experiment to these HUR transgenic mice, here we provide the inflammatory agonist, which is going to obviously promote or expression of many inflammatory mediators, and these over time will be able to potentiate tumorigenesis. When HUR is overexpressed, you see probably on average about a 2-fold increase in the number of colonic tumors. So I just want to summarize very briefly here a cartoon version of some of the data that we presented. Unfortunately, I didn't have a chance to present more on the mechanistic aspects of this. But normal colonic epithelium, this TTP mRNA decay factor is present. And this can bind the genes that are being expressed in the epithelium. Remember, the colonic epithelium is under a state of mild inflammation pretty much most of the time. And it can bind to RNAs that contain these AUH elements, bring them over to P-bodies in the cells where they can be, those RNAs can be degraded. And thus we can control gene expression. Whereas during tumorigenesis here, loss of these mRNAs to be targeted for degradation is occurring. And one thing we know that's happening is this RNA stability protein, HUR, is being upregulated and TTP expression is being lost. And I didn't have time to show the data here, but this loss of the TTP expression actually inhibits the levels of P-bodies that are being made in these tumor cells. And so through the silencing of TTP and the upregulation of HUR, you get stabilization of these RNAs, allowing for overexpression of the potentially pathogenic genes. And so the people who are currently in my lab, Fernando Blanco, Ashley Moore, Cyril Sobolowski, and Gretchen Dollar, have all played valuable roles in this. I'd like to give let you know that Fernando here is presenting a poster, and that poster will be on looking at some inhibitors of this HUR, using HUR as a therapeutic target. Some of my collaborators at Vanderbilt and former members of the lab. Thank you very much.
So, what is the expression level of TTP and HUR in other tissues?
With regard to HUR, that's been paneled in a number of different tissue tumor types, primarily of epithelial origin, and there is a strong correlation to the upregulation of HUR. Now, with respect to TTP levels, that has not been as exhaustively found, but at least in some tissue types, and our work in the lab, you also see this dramatic downregulation of TTP. So again, this is something that you don't see just one being upregulated, you see upregulation and downregulation.
you look at the effect of HUR expression in the context of APC mutation in colorectal cancer tumorigenesis. What about the background of other mutations, downstream mutations such as KRAS or P52?
Well, and that's a limiting factor with the APC min mouse because typically these mice do not live long enough, and this is the hypothesis, that they don't live long enough to generate adenocarcinomas. Okay, and so primarily the data that I'm showing you here is at 15 weeks of mice age, and that's usually at the point when they're starting to get pretty sick. But the majority, or all of those tumors that I demonstrate, are larger adenomas.
But you can use the biopsy, human biopsy, to just look at the mutation and the expression, right?
I'm sorry, I didn't.
Well, if you have a human biopsy.
Right. Okay, so we didn't, okay, I understand what you're asking. We did not go back, based on our tissue analysis, we did not go back and correlate to RAS or P53 or TGF-beta mutation per se, primarily because these were limited samples that were commercially available and from tissue arrays. So things were limited in that particular context. So, but that's a great point, and that's something in more of our cell line studies we're trying to correlate what is the genetic status of the cell, and are these certain known mutations a contributing factor? Yeah.
I have to ask you a comment. From a drug discovery point of view, how do you think it's possible to distinguish between the cancer-promoting HUR and the physiological HUR?
So I'll mention, please go to Fernando's poster. He has data actually illustrating that. But in essence, we can take a certain inhibitor of HUR, one that acts to inhibit cytoplasmic translocation. And we can look at a normal cell, and we can look at a tumor cell. And the main difference between those cells is looking at the levels of HUR, one being having HUR in the cytoplasm. Now, the sensitivity of those two cells to these drugs are dramatically different, probably on the order of 3 to 4-fold with regard to an IC50 pertaining to drug. So it's not to say that these compounds don't impact a normal cell, but the efficacy of them acting on a normal cell is much lower than in a tumor cell.
One last question.
I noticed that you had the highest differences between normal and tumor in the early stage tumors. Can you explain that?
Oh, yeah, that we've never looked at. I guess I can only say that maybe based on numbers. If we were to do a higher number of tumor samples, in that case, those numbers may start to come down a little bit. But right now, yeah, it could be. And the problem when you want to start looking at mechanism, obviously with tumor tissues, you're limited in that regard. And whether or not and a cell line is obviously from either a metastasis or a late-stage adenocarcinoma. So, and this kind of gets back to this other gentleman's question where you want to ask, okay, are there differences in the mutation capacity? And could there be activation of the silencer or something like that would downregulate things? But we've never looked in particular, but that is a very interesting point.
Thank you very much. We're slightly behind the schedule, so let's take, instead of 15 minutes, 10 minutes break.